My name is Per Venge. I am a professor emeritus of University of Uppsala and also a consultant at the University Hospital of Uppsala. Presently, I'm the CEO of Diagnostics Development. My research interest during many years that has been to purify novel proteins from neutrophils and eosinophils and also to make assays for these for diagnostic purposes. What I will try to do today, that is to show you the many diagnostic potentials of one of these proteins, that is Human Neutrophil Lipocalin HNL. HNL has many clinical applications: viral, bacterial distinction, sepsis monitoring, acute kidney injury diagnosis, can be used in COVID for prediction, and also in inflammatory bowel disease and irritable bowel syndrome. But before we start to discuss these clinical applications, there is something you have to understand, and that is that HNL exists in several molecular forms in various fluids of the human body. In fact, we have both monomeric forms, dimeric forms, and heteromeric forms. And this is illustrated in this simply by making Western blotting of urines and neutrophil release products and epithelial cells. The interesting thing is that the monomeric HNL is produced by epithelial cells as well as by neutrophils, whereas, and this is very important, dimeric HNL is produced only by neutrophils. This slide is an example of the differences of these various forms. We have collected plasma and serum from 2 patients with different kinds of infections, that is influenza A and bacterial infections. And what you can see here is that in the influenza A patient, both in plasma and serum, you have the monomeric, you have the dimeric, and also some heteromeric HNL. Whereas in the bacterial disease, the dominating protein here is actually the dimeric, and that is what is produced by the neutrophils. By means of our monoclonal antibodies and polyclonal antibodies, we have been able to combine these in many different ways, which has enabled us to make unique assays of HNL and its different forms. One of the more spectacular variants of this is the making of an ELISA that uniquely measures the HNL dimer. And in this slide, I show you with the blue the dimeric HNL protein measured by this ELISA, and you can see there's a nice curve. Whereas if we try to measure the monomeric HNL with this ELISA, we fail. There is no signal at all. So to summarize a bit of HNL, we can say that HNL, Human Neutrophil Lipocalin, is actually, it has many different names, NGAL, Lipocalin 2, and even others. But it's the same gene product. And when we purified HNL, at the same time, the group of Niels Borregaard in Copenhagen purified this protein and called it NGAL. So, as I said, it exists in several molecular forms in plasma and other biological fluids. And the cellular origins of HNL are neutrophils and epithelial cells. The dimeric form of HNL is unique to neutrophils, whereas the monomeric form originates from both cell types. And in most cases, it's not preformed in the epithelial cells, but it has to be induced by various mechanisms. And what I also said is that the combination of HNL maps and PABs gives rise to unique possibilities to construct unique immunoassays with different clinical diagnostic targets. So let us turn into the clinical applications. First of all, and to me still is the most important clinical application, that is the distinction between bacterial and viral disease. And for that purpose, we can measure HNL in serum, in plasma, or after whole blood activation. Whole blood activation means that we take 5-10 microliter of whole blood and then put an activator of neutrophils into that, and then we measure what is released from the neutrophils. And these different assays, they are actually superior diagnostic tools to distinguish between bacterial and viral infections, and all with the aim to reduce antibiotics abuse. There is a catch here with serum measurements, since obtaining serum for the measurement of HNL requires actually strict standardization of blood sampling procedure as to time and temperature. If you are not very strict, you can have both falsely high and falsely low levels. And for that purpose, as I already mentioned, we developed a whole blood activation procedure that completely eliminates this problem. And the good thing about this is that when we measure HNL released in the whole blood activation procedure, then it's very well correlated to serum obtained in a strict standardized fashion. One example of serum measurements is this one, where we have obtained a number of patients with acute infections caused by virus or bacteria. The samples were obtained before antibiotics treatment, and the patient's diagnosis were confirmed by microbiology and PCR, etc. And as you can see here in the left panel, in the middle there, then it's patients with bacterial infections, and to the left and to the right you have results from patients with viral infection and healthy subjects. We compared this also to other current measures, that is CRP and neutrophil counts, and to the right you can see ROC curves of these assays and their clinical performances. And obviously the blue line here representing the HNL assay is very close to the upper left corner, which indicates that it might be ideal for this kind of discrimination. And below you can see also the figures that we obtain with HNL a negative predictive value of 98% and a positive predictive value of 93%, and obviously significantly different from other currently used assays. I will also show you another very small study on bacterial and viral pneumonia in a hospitalized population, because this gives us an idea of how we can use, not only for diagnosis and distinction, this HNL analyzer. We can also monitor successful antibiotics treatment. And here you can see that if patients had taken their antibiotics 3-4 days before they were admitted to the hospital, then the levels were obviously lower and in some cases normal. So that was serum results. Now I will show you some results from the whole blood activation procedure. And here I will show you two things that are quite interesting. First of all, if we just base the diagnosis on clinical data, including neutrophil counts and CRP, then we do not have any very good distinction between CRP and HNL, non-significant, although HNL and PCT, they are distinct. But if we base the diagnosis on confirmed etiology, that is bacterial growth and PCR of virus, etc., then you can see that the blue line here, which represents HNL, actually goes up and approaches again the upper left corner. And below you can see the statistics here that HNL now distinguishes itself against CRP with a high p-value as HNL does when it comes to PCT. So this is very important to understand, that when we do evaluations of these kinds of marker, we need to have confirmed etiology and diagnosis of the patients. We also measured a number of other plasma-based markers and combined them with whole blood activated HNL, because after all, we didn't obtain 100% separation, distinction between bacterial and viral disease. So we asked ourselves, can we get even closer to 100% distinction? CRP only in this material, I had an area on the ROC curve of about 75%, and the whole blood system had an area under the ROC curve of more than 90%. But you can see that the red bar here, there we have added trail, and actually we approach 100%. We do not show this in this particular slide, but with IP10 we could show a similar thing. We also made a ranking of the individual assays here for the distinction and discrimination between bacterial and viral respiratory infections. And it's again very important to emphasize that all infections were confirmed by the etiology. And here you can see that serum HNL is actually in the top. But as I said before, it requires strict standardized blood sampling and preparation of the blood to obtain serum, a reliable serum. Blood activated HNL comes next, and then comes neutrophils, trail, IP10, plasma, HNL, CRP, and far down below PCT and heparin binding protein and thymidine kinase. So also as individual assays, HNL measured in either way are the superior ones. Next, that is sepsis. And for sepsis, our favorite assay is the HNL dimer. That is the HNL assay that reflects the involvement of neutrophils. We can also use whole blood HNL in whole blood after activation to distinguish from other diagnoses, predict the outcome, but most of all, and most importantly, for therapy monitoring. And here's one example of that. Here you can see that those patients with sepsis, they have much higher levels than those patients with a bacterial pneumonia or those patients with a viral pneumonia and influenza. And of course, they were all elevated as compared to normals. But if we made ROC curves and if we measured PCT, procalcitonin, in the same material, we could not show actually that HNL was superior to procalcitonin in the sepsis versus viral influenza distinction, nor could we show that it separated the sepsis from bacterial better than PCT, our HNL dimer. And if we look at some of the other biomarkers, you can again see that the area under the curve of the HNL variants are above 0.8, as is PCT, whereas the other assays, calprotectin, heparin binding protein, CRP, are clearly below that. One important thing when it comes to sepsis, that is prediction. How can we know which patient will end up with a severe sepsis or even die from their sepsis, and who won't? SOFA score is a score of the severity of sepsis, and if you have SOFA above 7 and up to even to 14, you are certainly about to die, then you have a very severe sepsis. And what was interesting is that if we measured the HNL dimer in a cohort of patients and related that to SOFA score, then you can see that those with SOFA more than 7 had significantly higher levels at the day of sepsis diagnosis. This is long before they actually developed the severe signs of sepsis and even died. This was not the case for PCT, as you see in the right panel, where there is really no significant difference between the two groups with SOFA more or less than 7. One of the very important things when it comes to sepsis, that is monitoring of antibiotics treatment. Because you want to know when you start to treat a patient with sepsis, does this antibiotic work or not? And that is a very tricky thing because these patients are multi-sick and it's a very complicated clinical situation they are living in. And to our surprise and to our really, we were quite happy when we saw that the dimeric HNL was reduced within 24 hours after adequate antibiotics and stayed so and became even lower later on. This is actually quite unique, because when you look at other assays, PCT, for example, which has been used for this purpose, then we couldn't see that PCT was reduced earlier than 4 days after initiation of adequate antibiotics. The total levels of HNL was not altered, CRP was not affected, calprotectin was not affected, but the HNL dimer was very rapidly reduced as a consequence of antibiotics treatment. And this can also be shown for the individual septic patients. Here you have on the left the dimeric HNL, which very regularly is reduced after adequate antibiotics, whereas on the right side you can see procalcitonin, which does not show this kind of regular reduction. Some goes up and some goes down. There is no significant change. Now, acute kidney injury. and COVID. That's another major indication for HNL. And it's not so surprising for you, maybe, because, as I said initially, HNL is the same gene product as NGAL. So we measure HNL in plasma and in urine, and we have shown that it's a tool for diagnosis, for monitoring, and also for prediction. This slide is quite interesting, and this illustrates what I talked about before. Here we have 5 different HNL assays. Yeah, one assay is actually the NGAL assay from R&D, but they are supposed to measure the same thing, that is HNL or NGAL in plasma. But what you can see that by changing the conformation of these assays as to antibodies, catching antibodies, detecting antibodies, then these assays actually pick up very different levels of HNL. And the one that is most striking here is the format 763-8F that actually picks up most of all these, whereas the HNL dimer picks up 10 times less. And we have used these, compared these assays in patients with acute kidney injury. And this is from a COVID patient cohort at their admission to the ICU. And they were, of course, admitted to the ICU because they had respiratory problems, and some also had signs of organ failure, such as acute kidney injury. And what you see here in the left panel is that this assay 763-8F actually is very closely related to KDIGO. KDIGO is a collective algorithm of the severity of kidney injury. So KDIGO3 are the ones who are the most severely ill patients. But you couldn't see that with NGAL at day 1. And if you transfer this into ROC curves again, then you can see that the 763-8F actually predicted survival fairly good, not perfect, an area under the curve of 0.76, but significantly different from NGAL and also from the dimer assay. The severity of AKI actually, there the dimer and the 763-8F assays were similarly related, but again, NGAL was less related to severe AKI. Then these patients were sampled day 3, and of course they had a progression in their organ failures, such as the kidney. And you can see this again, that those with KDIGO3, they had very highly raised levels of HNL. And if we again transfer this into ROC curves, then you can see that as to survival prediction, now NGAL is not different from HNL, whereas still the severity of AKI is more closely related to the levels of HNL total 763-8F as opposed to NGAL. Then I would like to show you this, because I think this data is very intriguing. At the time when COVID was reasonably young, we understood that the COVID infection did something to the body that we didn't recognize from other infections. And the idea and the findings suggested that maybe the neutrophils are involved in this process as well. And we set out to investigate this in a fairly large COVID cohort. And since we had the tools to measure neutrophil involvement, either by plasma levels of HNL dimer or by activation of whole blood, we looked at this then. And what you can see here is, it may be very difficult to see, but we had a certain cutoff and related or predicted severe AKI stage 3 by 40% in those subjects who was above this cutoff. And when we looked at the activated whole blood HNL, then we predicted by 60% those who actually developed AKI stage 3 during these 50 days of observations. The last clinical application I would like to talk to you about, that is HNL in inflammatory bowel disease and in irritable bowel syndrome. You may know that irritable bowel syndrome is maybe the most common complaint of all human beings. It has been estimated that IBS, from its most severe form to its mildest forms, affect 20% of the humanity. But nothing is known, or very little is known, about the pathophysiology of IBS. However, when we do this, we measure HNL in feces. And for that, it's potentially useful tool for both diagnosis and for monitoring of IBS and IBD. Here you can see that when we measure with our HNL PAB765 assay, then those with Crohn's disease, CD, have significantly higher levels than healthy subjects, as do those with ulcerative colitis. And that is not unexpected. We know that these two diseases are driven by neutrophils. The surprising thing is that IBS is much higher than the healthy subjects. We made ROC curves, and you can see that distinction between IBD and healthy persons. Calprotectin is what is being used today, but the HNL, PAB765, and even MPO seem to be slightly better for this. When it comes to distinction between IBS and healthy persons, then calprotectin does nothing. It's similar in between these two cohorts. EDN, the eosinophil-derived neurotoxin, is actually significantly higher, indicating an eosinophil component in this disease. But what is really striking, that is HNL, which is at least twofold higher in IBS than in healthy subjects, and MPO is not at all. And if you make ROC curve, you can see that the only assay that really shines here, that is HNL PAB765, whatever that means. And also, we have a relation to subtypes of IBS. So the constipation type, the diarrheal type, the mixed type, they have higher levels of HNL. So finally, the many clinical applications of HNL, they are bacterial viral, superior distinction by whole blood activation of serum, it's sepsis, prediction and therapy monitoring by plasma HNL dimer. It's acute kidney injury, monitoring and outcome prediction by plasma and urine. It's COVID outcome prediction and diagnosis of organ failure by plasma, urine, or whole blood activation. And IBD-IBS, identification of unique activities in IBS by feces analysis. And probably much more is to be found. Thank you very much.